in the today's class we'll study about the dermal tissue system. The name itself indicates the dermal nothing but epidermis, nothing but outermost layer. So usually the dermal tissue system broadly in the plants comprises of three types of a tissue system. One is epidermis, followed by the epidermis comprise stomata, and one more specialized epidermis. So in the today's class we'll understand or we'll study about the two important parts or tissue systems which are exclusively found only in the epidermis, not in other places. So you might agree with me that the stomata and the trichomes are the two components which we can see only in the epidermal region, not anywhere else, right? So in that, today we'll talk about the tissue system. In that, exclusively about the dermal tissue system. And before that, I would like to give some little information about the tissue system. I mean, what do you mean of tissue system? So tissue, we already know that tissue is nothing but a what we call organization of structurally similar cells, performing a different number of cells performing general function irrespective of their position as well as their continuity. Nothing but their position may be different and their function for a different period will be the same. So all these together will be considered as a tissue system. And in that, today we are talking about the dermal or epidermal tissue system. Again, I would like to give one more time, that is what you mean of dermal tissue or the epidermal tissue system. Nothing but the outermost protective covering of the plant body will be considered as the dermal tissue or epidermal tissue system. And usually this epidermal tissue system grown will be represented only, it will be represented at the time of plant reaches, when the plant at the primary plant body or a primary plant growth. So what happens usually when the embryo start dividing, when we're talking about the dermatogen layer or a 16 cell stage, there you are going to see the dermatogen formation. Nothing but this is what the outermost layer forming meristemations are going to form the epidermis, nothing but which acts as a protective layer for the microorganisms and also to stop the different mechanism. Nothing but a loss of water molecules may be there, and also invading mechanical injury may be there, and all the protective mechanism will be rendered by the epidermis. What happens in the primary during the primary growth, we are prominently we can see the formation of epidermis. So when we talk about the secondary growth of a plant body, particularly the epidermis will be completely eradicated or completely removed by the another type of a dermal system, that is we are going to see the periderm. Nothing but the secondary growth during the secondary growth of the plant body, there we are going to see the replacement of epidermis with the periderm. This periderm acts as the outermost protective layer, and this usually contains the cork cells. These cork cells are the buoyant in nature, nothing but, so when you take the cork, I mean the when you just go and take any woody plants, if it is having more than 10, which is exhibiting the secondary growth, you just take the cork and put it in the water. So you are going to see it is going to float on the water. So that buoyancy is just because of the cork cells. This cork cell containing the periderm will act as a new protective layer. Nothing but the dermal tissue system, the epidermis, which exhibit the two phases according to the growth of a plant body. During the primary growth of the plant body, the prominent epidermal cells will be seen, and which acts as a protective layer, outer covering layer, which will be protecting in nature. When the plant at the secondary growth stage, where we are going to see the epidermal cells completely, epidermal cells will be replaced by the peridermal cells, and these peridermal cells which contains the secondary growth cells, exclusively cork cells, and they act as the protective tissue for the plant during the secondary growth. So in that, let me talk about, today we'll talk about the two components of the epidermal tissue system. One is the stomata, another one is the trichomes. The first now we'll talk about, okay. So usually the leaf contains stomata. Usually, even the stem also contains the stomata. Where they are absent, where the roots lack the stomata. Why? Because again, the roots which are not exposed to the sunlight, because some of the external factors determines the formation and the functioning of a stomata. Okay, now. So usually the one more big factor, external factor, that is the sunlight, which determines the position as well. I mean the function as well as the what we call the location of the stomata on the plant body. Usually the leaves which are exposed greater towards the sunlight, so the leaves will be having more number of stomata followed by the stem. I mean the young stem or a primary growth stem. These stomata are the minute pores which are quite disc-like, I mean, which are quite different than the conventional epidermal cells that you have seen already in the UG. That so when you take a section or when you talk about the stomatal index, there you will come across the cells, the leaf cells, which are having some quite structurally different cells, and those which looks like the kidney or bean shaped cells which are surrounding by the accessory cells. Am I audible?
Yes,
Yes.
yes.
Right. Now coming to the structure of stomata, is the stomata are the minute pores which occur exclusively on the epidermis of a plant body, that is on the stem, more number in the leaves. Each stomata is remain surrounded by. Structure. I would, though it is basic, I would like to give some little information about the stomata. So here, the stoma remains surrounded by two kidney or bean-shaped cells, and those will be considered as the God cells. Nothing but the main function of the stomata is based on the God cells. Nothing but the opening and closing of a God cells, which determines the functioning capacity of a stomata. Where the stomata usually present on any part of the plant body, as I already told you, except on roots. Why? Because they are the external factors which determines the function and the location of a stomata. Usually, the external factor, one more, the best example for the external factor is the sunlight, where sunlight, the roots will never get exposed to the sunlight. Because of that, they lack stomata. Epidermal cells bordering the guard cells are called as accessory cells or subsidiary cells. But where the guard cells, which are quite different than the conventional epidermal cells, and these guard cells, one more structural feature of the stomata is they contains 2, 3, 4, or 5 number of accessory cells or subsidiary cells, and we'll show you the images of the stomata and how they are structurally different, the accessory as well as subsidiary cells. Nothing but usually what happens, the guard cells will remain same with quite or a rough variation can be seen in the stomata, structure of stomata in more monocot, dicot, and even in the gymnosperms and also the lower organisms. But the accessory or subsidiary cells pattern will vary and will determine the different types of the stomata and their function. And usually what happens, these God cells are living because they contain the active protoplasm and they also contain the chloroplast in them. So because of containing the chloroplast, they can even conduct the photosynthesis. Right? Nothing but they are not directly involved in the food production of food, but because of containing the chloroplast in them, so the activity of closing and opening of the stomata will be rendered very fast. And will detailed function of the stomata will be studied in the coming slides. And stomata, the arrangement of stomata broadly, where we can see in the dicotyledons as well as the monocotyledons containing the stomata. Even the lower organisms such as the gymnosperms also contain the stomata, where like algae and the fungi and some of the primitive non-vascular plants, they doesn't contains the stomata at all. Where when we talk about the occurrence of a stomata, where we can see the sunken stomata, nothing but a very primitive stomata can be seen only in the gymnosperms. Later on, there is an during the course of evolution in the monocotyledons as well as dicotyledons, where we can see there is a morphological as well as the functional variation, a quite distant variation can be seen in the stomata in the dicotyledon as well as the monocotyledon. Now coming to the distribution of stomata in the dicotyledons is quite scattered, nothing but they are irregularly distributed here and there. There is no actual pattern of distribution in the dicotyledons in the leaves of the stomata. Where in the monocotyledons they are quite arranged in the parallel pattern. Nothing but if this is what a leaf in the long when you take about a long leaf, there you can see they are arranged one below each other. So we'll show you the images of that, how they are arranged in the monocotyledons. But remember that the irregular distribution or scattered distribution of a stomata can be seen in the dicotyledons, where the parallel arrangement in the form of rows, the parallel arrangement of the stomata can be seen in the monocotyledon leaf. So, And one more, a few more important characteristic feature of a stomata is usually the stomata remain closed in the night and open in the daytime. Can you during the light, during the daytime? I mean, why I'm telling light? Okay, now, during the daytime, we are going to see the plants can get the light. So that with the utilization of a light, they can go for the conduction of the photosynthesis. During that time, we can come across the exchange of a gases. So for to have an exchange of a gases, the stomata will remain open during the daytime. So during the night, I mean in the absence of a light, I'm not telling that in the night, usually night will be dark, and nothing but the absence of light during the night or in the absence of a light, they remain closed. One more important feature is that during the scarcity of water or during the lack of water, with I mean during the drought stress condition also, the stomata remain closed. Why am I telling? Because the function of, as I already told you, that the function of stomata is completely determined by some of the external factors. It may be sunlight, it may be light, normal light, it may be even the water scarcity. Okay, na? So during what happens, because it is with the help of water and the light, it is going to produce the sucrose. I mean, it is going to produce the food material. So what happens if in the absence of a light and in the absence of the water and in the absence of in the lack of or in the lack of or a scarcity of water, the stomata will be closed. So why it is closed? To avoid the loss of water molecule or to avoid the loss of gaseous exchange, to avoid the loss of mineral moisture. So they are closed in the night. Generally, it will be regarded as they are closed in the night. They are open in the daytime. And the number of stomata, the occurrence and the number of stomata on the surface of a leaf varies. Nothing but the number of stomata may be hundreds to the thousand per square centimeter. Nothing but so when you observe it under the microscope, you will come to know. So that's what to calculate the number of stomata. That is what the stomatal index. We have some of the experiment, right? So during that time, we'll come to know that per square centimeter, how many number of stomata are there. So in some of the plants, we can come across hundreds of stomata. In some plants, the leaves, a single leaf may have thousand number of stomata. So again, based on the physiological function of a leaf as well as a plant, we can come across a varied number of stomata. And one more point is that usually in the terrestrial plants or mesophytic plants, the number of stomata are more in the lower surface than in the upper surface. Why in the mesophytes? I mean the terrestrial plants usually they have the stomata. The number of stomata are restricted more in the lower surface. It doesn't mean that the upper surface of a leaf doesn't contain the stomata, but their number is less. Why? Because the upper surface of a leaf will be exposed greater towards the sunlight than the lower surface. So the gases exchange should be happened from the lower surface, greater in quantity. So the lower surface of a terrestrial and as well as, I mean the mesophytic plants contains more number of stomata in the lower surface. So where in case of hydrophytes, their lower surface of a stomata, lower surface of a leaf doesn't contain stomata at all because their lower surface is directly connected with the water. So whatever the exchange of gases should occur, it should occur from the upper surface itself. Okay, na? Where in case of the in the xerophytes, can anyone tell me where the xerophyte doesn't have a peculiar leaf? Because I mean, leaves are modified into thorns, leaves are modified into spines and filler plates. Different patterns of variation can be seen in the xerophyte. So usually the stem of the xerophyte contains the stomata, and there again because of there is no we cannot classify the stem of a xerophyte into adaxial as well as I mean the uh, uh, dorsal as well as ventral surface. So the stem of the xerophyte, there again distribution of a stomata cannot be classified as the lower as well as upper. Okay, na? So this is the first image which indicates that the stomata containing the granules. Can you all see the first and second image? Can you all see these the granule structures? These are the chloroplast, and this is what the closed one, and this is
Yes,
what
sir.
the open one. So this is
Yes,
the
sir.
first image. Is the closed stomata, and this is what the open stomata. Got it?
Yes, sir.
Now, coming to the based on the structural variation in the stomata, we have got functionally all the stomata perform the same function. That is what the gaseous exchange. Structurally, we have got different types of stomata, and exclusively the dicotyledons. we are going to see the four types of stomata. One is anamozytic, anisocytic, diacytic, and parasitic. Again, we'll repeat again. Anamozytic types of stomata, anisocytic type of stomata, diacytic stomata, and parasitic stomata. Where in case of monocotyledons, we are going to see only one structural similar types of stomata in any type of a monocotyledons. So if you go and start taking the stomata of from poaceae of the grass and a Oryza sativa, and you can take another monocotyledon, there you are going to see the structurally they are same, but their number may vary. But the structurally monocotyledons contains the gramineaceous type. They contain a parallelly arranged stomata, and that has been named as gramineaceous type. Where in case of dicotyledons, we are going to see the varied structures. That we have got four types. And let us talk about the first one, that is anamozytic type of stomata. So it has been said as ranunculaceous type of stomata. So nothing but it's again this type of stomata can be seen in what to call in the primitive. Broadly, it has been said that hypothetically that in the lower or a primitive angiosperm, there is this particular type of stomata can be seen. Hence, the name has been given as ranunculaceous, because ranunculus or ranunculaceae is a primitive family that comes in when we talk about the evolutionary phylogenetic classification of plants. Ranunculaceae comes as a primitive angiosperm. So in generally, it has been said like that. But the alternate name for the anamozytic type of stomata is again as ranunculaceous type of a stomata, and it has been also said as type A type of stomata. Well, now the what do you mean of anamozytic? Anamozytic nothing but irregularly cellular. Nothing but the subsidiary cells, as I already told you that based on the subsidiary or based on the accessory cells, the stomata have been classified structurally different types. Where here we are going to see the irregularly shaped subsidiary cells. Nothing but one subsidiary cell will not be, for example, it may be having 4 or 5 number of subsidiary cells or the epidermal cells, and they are quite different than the remaining. For example, let us see here we have got 4 accessory cells, also 4 subsidiary cells. Right? Just see one subsidiary cell is not matching with this structurally matching with another subsidiary cell. One is quite small, one is quite big, and just see even the perforations. I think, but the troughs here what we are going to see are quite different. So the anamozytic type of stomata, where the 2 guard cells are having accessory or subsidiary cells, quite number maybe 4 or 5, but all they are quite structurally dissimilar, quite alike than each one. Those type of stomata can be said as anamozytic type. Anisocytic type of stomata, and it has been said as type B. Where anisocytic type of stomata, here what do you mean of anisocytic? The unequal celled. Here we are going to see the stomata which is surrounded by the accessory or subsidiary cells. Their number is 3. Nothing but there we have seen an anamozytic. There we have seen 4 or 5 subsidiary cells which are quite unlike each other. Here we are going to see exclusively 3 accessory cells or 3 subsidiary cells, which are out of that one subsidiary cells is quite smaller than other 2. So if you want, you can check here. So this is what the microscopic image of the subsidiary, pardon, the cruciferous or anisocytic type of stomata. Here you can see that 1, 2, 3 are there. 1, 2, 3 are there. So just see one is quite smaller than the remaining 2. Right? The structurally, here we are not going to see any troughs, any perforations, or any outgrowth like that. Here they are just smooth surfaced one. But out of the 3, one subsidiary cell is quite smaller than the remaining 2. And those type of stomata will be considered as anisocytic or cruciferous type of stomata. Now, the next we have got the third one is that rubiaceous type or the parasitic type of stomata. Again, type C type of stomata. Parasitic nothing but the subsidiary cells are parallelly arranged to the guard cells. Nothing but if the guard cells are like this, in the long axis, they are again, the subsidiary cells are arranged parallel to each other. And that you can see here in the image that the 2 subsidiary cells, here you can see the 2 guard cells which are arranged parallel to that. One subsidiary cell overlapping the another guard cell, and here you can see the guard cell which has been overlapped by the another guard cell. Nothing but the stomata where containing God cells which are parallelly overlapping or parallel to the long axis of the pore of a God cell. So you can see the pore and they are parallelly arranged. So this is what the microscopic image. Just here you can see that the God cell is here parallel arranged like this, and again God cell parallel arranged. Here the God cell parallelly arranged. Anyone able to see this one? Is it clear? Is it clear? And now coming to the fourth one, that is a caryophyllaceous type of stomata or a diacytic type of stomata. So now the diacytic type of stomata is quite contradictory to the parasitic. Nothing but where in the parasitic stomata, there we see that they are arranged parallel to the long axis of the pore. Here, the diacytic type, we are going to see the cross-cellular type of stomata. Nothing but here the stomata which has got perpendicularly arranged to the God cells or the right angled arrangement of a subsidiary cell to the common wall of this God cell. So you just see here, its stomata is there and one God cell which has been perpendicularly arranged and another God cell is also perpendicularly arranged. Here also you can see that this is what a stomata where the perpendicular arrangement of the stomata, the God cells. Can you all find the difference? Anamozytic, anisozytic, parasitic, and diacytic. Is it clear? All the four now? Shall
Yes, sir.
Okay. Fine. Now coming to the gramineous stuff. As I already told you that in the monocotyledons where we are going to see the parallel arrangement of the stomata. Just see that is what the parallel arrangement of the stomata. Just see one stomata which is parallel arranged to the next. One stomata which is again parallel arranged to the next. Just 1, 2, 3, 4. Nothing but here you can see instead of irregular distribution of a stomata as in dicotyledons, in the monocotyledons we are going to see a parallel arranged stomata. Where in the gramineous type of a stomata, the what we call the guard cells are quite arranged in the middle portion and they are quite narrower than the ends. Nothing but they just looks like a dumbbell shape or the bone shape nothing but the ends of the stomata which will be quite swollen where when you come at the center we are going to see the compression and again at the end we are going to see the bigger the swollen portion. Because of that we can see that just like a dumbbell shape, and usually they are found commonly on the gramineous as well as the cyperaceous member of monocotyledons. And you can see here that at the center you have the God cell and the dumbbell shape and the subsidiary cells, the two number of subsidiary cells you can see here, right? Can you all see this? This is what the dumbbell shape which has been overlapped or arranged. Subsidiary cells are arranged at the along with the God cells. Here in the image you can see that.